Hello there, this is AGNR 131 Soil Science, and this is Laboratory 8, Exercise 1, Effects of Salts on Soils. I'm going to go ahead and read the introduction on this so you guys understand exactly what it is that we're going to be doing today in this part 1. As you know, we've been studying water movement in and through the soil in the last in past lab exercises. We've also been studying the effects of salt on plant growth and development. So what we're going to do today is we're going to actually look at how salt, different concentrations of salt, different kinds of salt will react with water movement under different soil textures. So what we've got going on here today is we've got 3 different soil textures, 3 different soils we've picked for you. One of them is a loamy sand. One of them is a silty clay loam. And the last one is a very nice classic loam. So we've got 4 different salt treatments. Our salt treatments here are just plain pure water, nothing in it at all. And then we have what we're calling a saline water, and that one has calcium, dominantly calcium added to the soil, and then some sodium. Then we have a sodic water, which is reversed. It has a little bit of calcium and a lot of sodium. And then we have what we are calling a saline sodic treatment, or saline sodic water, and that one has it has a lot more of both. It has a fair amount of calcium, but almost 4 times as much sodium in it. So I'm going to go ahead and provide you with the electrical conductivity of these, and I might even give you the SAR, and you can look up those terms if you've forgotten what they mean. So you'll have a table that shows these treatments in your lab exercise handout. And I'm also going to put the data table there that will put the results of what we're going to do today. So let me just sort of summarize again. 4 different treatments of salts, no salts, and then dominantly calcium, a little bit of sodium, and then dominantly sodium, a little bit of calcium, and then a lot of both. Okay? And we're going to actually measure how on these 3 different soils, we're going to put each one of those treatments, and we're going to run them 3 times. So we're not just going to take the results of 1 repetition, but we're going to run them 3 times. So what we'll do is we'll have, and I've got some prepared over here. I can kind of show you that we've got already put our samples in there, 50 grams of soil, and I've actually got filter papers in here, and I've just wet them down so they'll lay flat, because I don't want the water to, when I pour it in here, to run underneath that filter paper and go straight down into the flask underneath it. So each one of the filter papers has been wetted, and I've already put soil in this one for our first run of this is the silty clay loam, soil C on this one, silty clay loam. And we're going to go ahead and pause here and go ahead and put the rest of them in here. And then what I'll do once we get these things all loaded up for run 1, I'll explain what's next. So here we've got all of our Buchner funnels sitting in their flasks, all filled up with the different soil samples. So we've got one set here of 4 funnels, and then obviously the middle set of 4, and then the last 4 sets. So I've got them on this side. You can't see it. I'll turn them around, but I've got them just to keep track. I've got them numbered 1, 2, 3, 4. So each soil, and remember the first one here, soil A, was a loam. Soil B was a loamy sand, and soil C is a silty clay loam. I'll put that on your data sheet so if you don't forget that. But I've got all my treatments labeled 1, 2, 3, 4 here. So what I'm just going to show you, so here's our first treatment. This is what we're going to do all the way around. We don't really need to video it, but I wanted to show you for the first one what we're going to do. So the first treatment is just deionized water. So every number 1, all 3 soils, one of the 4 Buchner funnels is going to get the treatment of deionized water, right? So all we're going to do right now is we're just preparing the soil. So I'm going to carefully just pour this water in around the soil. I'm not going to pour it in fast, because I just kind of want it to sink in. I don't want it to displace any of the soil material on top of the Buchner funnel any more than I have to. I'm just going to carefully put that in. So we're going to repeat that process. I'm going to fill up deionized water in the other 2 sample sets, and then I'm going to go back and put each one of the different treatments in that. No sense for me videoing that, but when we come back and get all those preparing the soil for the salts, if you will. That's actually what we're doing. We're applying our treatments to the soils right now. So then after that, what we're going to do is we're going to go ahead and see what happens when we just maintain water on top of that. We're going to maintain just regular water on top of this after we've already put our treatments in here of different salt solutions. So that's going to have some effect on the soils. We don't know what it's going to do yet, or how the different textures will react to those different salt, calcium, and sodium solutions. But that's kind of what we're going to measure. But the first step is just to go ahead and apply the appropriate treatments to each one of our soil samples. So we'll go ahead and do that and come back. Okay, so we've added all of our treatments here, and you can see that if I move these, you can see that some of them are already dripping through, and that's okay. That's okay. We haven't actually started the actual second part of the meat of the experiment, if you will. But it's okay if these things drip through, because we're still trying to just prepare the soil. So these treatments are actually working their way through the soil, and the soil sample is going to be affected then by the treatment. So it's okay for it to all drain through and sort of prepare the soil, if you will. So what we're going to do after this is we're going to go ahead and put water on top of this. We're going to go ahead and fill these beakers up, or these Buchner funnels up, with water, all to the same level. We're going to try to maintain what we call the same head, a constant head on them. And we're going to go back and forth, and as it drains through, we're going to go ahead and keep filling it up so it's pretty much almost at the rim here. So it like getting close to flowing over. So having done that, you can see we've tried to make these funnels as level as possible, so that the water will be sitting in there. So we're trying to eliminate any variables in our experiment. So we'll go ahead, let these things drip a little bit, then we'll come back with the water all filled up, and we'll talk about the next stage. So we've gone ahead and filled all these things up. We put in, to review, we have 50 grams of soil in each one. We added 100 mils of the treatment, and then on top of that we added 100 mils of the deionized water on top of each one of these treatments. And then what we did after that is, as it began to flow through, we maintained the head. And you can kind of see the water level is at the same height in all of these. So the same amount of pressure is pushing down on each sample. So I've got the loam right here, and I've got the loamy sand here in the middle, and the silty clay loam here at the last four. So there's the same amount of water pushing down on across the sample. So that's that same head. But now you can already see some interesting things are going on through here. There's some dirt in this first one. This one, it looks like there's a lot more water in number 1 and in number 3 on this. I don't even know if number 4 has even dripped through on this one at this point. So to remind you, the same thing has happened. The same thing is going on in each one of these soils. The only difference is the amount of calcium and sodium in the soil. The soil textures are the same. The amount of soil that we put in the sample was the same. It's just the salt content and type that's different. So we're going to let this run for a little bit, and I'll go down here. We'll just sort of zoom in on the camera. This is the loamy sand here in the middle. There's the loamy sand. This was the loam. This is the loamy sand. You can see, again, kind of an interesting treatment. You've got 1, 2, and 3 here. Even the colors are a little bit different. Number 4 again. This is number 4 here. I don't really see anything dripping through. Here's a silty clay loam. This has got a lot more clay in it than the other 2 textures. But I will draw your attention. This is the loamy sand, and this is the loam. Okay, loamy sand and loam. So I keep saying these things to you because I'm going to ask you questions about the different textures and what you're actually seeing here. Here I've got a silty clay loam, and I've got the 4 treatments: treatment 1, treatment 2, treatment 3, treatment 4. Again, refer to your lab handout to know which one those are. And look what's happening in through here. I see drops come through number 1. I see not much on number 2. Looks like number 3 is flowing pretty well. And number 4, I don't see anything really going on yet. So that's where we stand right now. I'm going to go back and make sure that the heads are all the same, that we have the same amount of water through here, and then we'll let this experiment run. And then when we're done, we'll come back and we'll look at the volumes. And then this will be run 1. This will be the data that you get. This will be the data for run 1 that you'll see on your lab report, your lab exercise. So we're just about ready to stop the whole experiment here, but before I do that and pour the volumes, the leachate down here, what we call the leachate, before I pour that into different beakers, so our different volumetric flasks, so you can see the relative amounts. What I wanted to show you was I wanted to bring attention to the fact that these are still full of water, or water or their treatment, but yet some of them aren't even moving. You can see here, number one, that would be the deionized water. I don't really see much happening. As a matter of fact, the only one I really see in sample one moving is the third treatment. That third treatment's the only one that's actually doing anything. The third treatment, again, is the sodic one. On that one. Over here, on this one here, it looks like the deionized treatment one. I see a little bit of movement that's still dripping, and I see not much else. There's a little bit of dripping there. It looks like number 4, which would be the saline sodic, it's still doing something. And then over here in this, and that would be the, actually I'm sorry, that's the loamy sand. The first one was the loam. This is the loamy sand here. And then you can see on this one, I'm getting a little bit of dripping going on all of them still, but nothing is really going very quickly. But still a little bit of dripping going on the silty clay loam on all of them. You can actually see there's obviously going to be a difference here. It looks like there is something going on with texture. Something, we'll figure it out. You guys will figure it out. Now we're going to go ahead and stop this at this point, and we're going to put the different quantities in there, and then we'll show that, and then we'll record that data, and that'll be presented to you as well. Again, this is still run 1, then we'll go ahead and collect run 2 and run 3, and then you'll get all that data. Okay, so we're on the final part of our first run of 3 for this particular experiment, and so I wanted to. This is a much more visual way of looking at it. So you can see here on the loam, you can see what's happened with the different treatments based on the amount of water that's actually come through I say water, but the soil solution that's actually come through each one of those different treatments. So here we go, treatment 1, which was the DI water, treatment 2, which was a saline soil, treatment 3, which was a sodic soil, and then treatment 4, just in this little tiny one, because you wouldn't have been able to see it in this larger graduated cylinder, the saline sodic. So that's the loam. Here we have over here, we have the same one. We have the same treatments in the same order with the loamy sand. I've got the DI water, and I've got the saline soil, I've got the sodic soil, and then I've got the saline sodic over here. And then finally the silty clay. The silty clay, you can see we've got the DI water here, silty clay loam, DI water here, saline here, sodic here, and then the saline sodic on that one. So that actually gives you a pretty good representation of how things work. So remember, what we're trying to ascertain here is how those different types of salts and different concentrations of salts actually will affect water movement through soil. And we added the different variable of looking at 3 quite diverse soil textures to see if there would be any comparison across soil textures. Now, we got to make a few more runs before we can really get some data that we can actually use. But here we go. You kind of see it visually on the first run. So we'll go ahead and run the other ones, and then you'll get all the data presented to you in your data sheet.